Good evening, everybody. Welcome back to Daily Lectures at New Age Cosmetic White at Tech Healthcare Consultancy. We are on the subject of molecular biotechnology and at the chapter dealing with directed mutagenesis and protein engineering. We are on the summary of the chapter. Now, onto the summary. It states the proper functioning of a protein is due to its conformation, which is a consequence of its amino acid sequence and subunit structure. Certain amino acids in a polypeptide chain play important roles in determining the specificity, thermostability, and other properties of a protein. Change in even a single nucleotide of the gene encoding a target protein can result in the incorporation of an amino acid that can either disrupt the normal activity or enhance a specific property of the protein. With the emergence of recombinant DNA technology, it has become possible to replace nucleotides of a cloned gene and to produce proteins with specific amino acids at defined sites. This procedure is called directed mutagenesis, and it can be performed in various ways. To change a particular amino acid within a protein, the target gene is first subcloned onto bacteriophage M13 DNA. The single-stranded form of this bacteriophage is then copied by using an oligonucleotide primer that is designed to introduce a specified nucleotide into the target gene. E. coli cells are transformed with the double-stranded M13, and some of the M13 bacteriophage progeny carry a variant of the cloned gene that contains the mutations. These bacteriophages identified the altered subject is subcloned into an expression vector, and the expressed protein is tested for activity. There are also plasmid and PCR-based strategies for introducing these kinds of changes into cloned genes. In many instances, the amino acid changes that might enhance a particular property of a target protein is not a known priority. In these cases, random mutagenesis, error-prone PCR, or DNA shuffling rather than oligonucleotide-directed mutagenesis is preferred. The choice of which amino acid to change is often based on knowledge of the role of particular amino acids in their function. And functional protein. This knowledge comes from the genetic studies of crystalline crystallographic data of three-dimensional organization of the protein. Specific sites or regions can be altered or combined to improve the thermostability, pH tolerance, specific allosteric regulation, cofactor requirements, and other properties of enzymes that are used in industrial processes. For example, thermostability has been enhanced by changing amino acids at two sites of the enzyme triose phosphate isomerase, and the protease sensitivity of streptokinase has been decreased by changing two lysine residues to glutamine. Not only are these approaches helpful in engineering new properties for existing proteins, but they can also be used to design unique enzymes. And now on to the review questions. Question number 1. What physical and chemical properties of enzymes are targets for enhancement by directed mutagenesis? 2. You have cloned a bacterial gene that is expressed in E. coli, and now you want to alter its activity. However, because of technical problems with the original M13 protocol, only a very small fraction of the mutagenized clones of this gene actually carry the modified gene. The vast majority of the clones contain the unaltered form of the cleared gene. How would you perform site-specific mutagenesis so that a much larger proportion of the clones have the desired mutations? You have isolated a gene from an enzyme that's expressed in E. coli. Describe how you would alter the catalytic activity of the enzyme. Assume that you know the DNA sequence of the gene, but do not know anything about which region of the enzyme are important for catalytic activity. 4. Discuss the advantages and disadvantages of oligonucleotide-directed mutagenesis using either bacteriophage M13 or PCR. Describe a strategy for oligonucleotide-directed mutagenesis that uses plasmid DNA containing the gene of interest. How can degenerate oligonucleotides be used to generate random mutations within a cloned DNA fragment? Describe a strategy for increasing the stability of a protein that has 1. No cysteine residues or 2. An odd number of cysteine residues. 8. How might replacing asparagine with another amino acid residue affect the stability of a protein? How might replacing asparage with another amino acid residue affect the stability of a protein? How can the cofactor requirements of an enzyme be altered? Number 10, describe how you would change the catalytic activity or substrate specificity of an enzyme whose gene you have isolated. Why would you want to do this? What is error-prone PCR and why is it useful? Next, outline 2 ways in which DNA shuffling may be used to generate hybrid genes. How can unusual amino acids be incorporated into proteins, thereby producing an altered form of the target protein? How would you engineer human pancreatic RNAs to be an anti-tumorigenic agent? How would you engineer streptokinase so that it was less sensitive to proteolytic digestion? How can the genes encoding a Fab fragment of a monoclonal antibody be modified so that the specificity of the antibody is altered? What is molecular breeding? How can this approach be used to simultaneously engineer several properties of a protein? Starting from a single enzyme coding gene, how can DNA shuffling be used to engineer an enzyme in which several separate and distinct properties have been modified? How would you use PCR-amplified oligonucleotide-directed mutagenesis to rate insertion mutants, deletion mutants? How would you generate random insertion deletion mutations in a target gene on a plasmid? What is non-homologous random recombination and how would you use it to generate modified enzymes? How would you engineer an enzyme with a higher level of activity towards one substrate to have a high level of activity for a different substrate and have a low level of activity toward the original substrate? How would you ensure that an entropeptidase cleave only at the target site within the problem of interest, protein of interest, and nowhere else? That ends the chapter on directed mutagenesis and the protein engineering. This lecture is a short one. It was done only for the review questions and the summary. I'm stopping it here now, and we join you with a new chapter, the next chapter, the number 9, titled Molecular Biotechnology and Microbial Systems, and we will be dealing with molecular diagnostics, protein therapeutics, nucleic acids as therapeutic agents, vaccines, synthesis of commercial products for recombinant microorganisms, bioremediation and biomass utilization, plant growth promoting bacteria, microbial insecticides, large-scale production proteins from recombinant microorganisms. Thank you. I'll join you in the next lecture.